So this being your first class, let me give you a short introduction about this introduction to IT, introduction to information technologies in this bioinformatics and genetics. So we are going to discuss about biological data. So that's the reason I asked you, do you have prior knowledge about biology? Okay, so we would be studying about DNA, RNA, chromosomes. Okay, so same thing, I'm going to repeat them. And then here in this particular class, we will discuss about genomes and then proteomes. Okay, so it's the syllabus is framed in such a way that half of our syllabus topics falls in bioinformatics, and then the second half falls into genetics. Okay, so it's more of biology. In biology, we are going to input computer engineering or maybe information technology. So this particular class will have a short intro for the biological data in digital symbol sequences, and then we discuss about the genomes. Okay, the various diversity features we will discuss about the size we discuss about, and then about the proteins structures as well as the proteomes. So actually proteins and proteomes are quite similar. Okay, right? So this is especially for you, my dear students and young researchers, and of course you can reach me at dr.christoananth@gmail.com. Okay, so no matter you are busy, no matter you are free, no matter you do anything, okay, at least take 1 minute prayer for each of the things. Okay, before eating food, before starting anything new, before starting a new business, before going to job, maybe after coming back home from university, don't forget to thank God, because it really helps you, and it is because of the abundant grace of God that we all do things positively. Okay, even if there are several hindrances, several situations can attack us 360-degree. Okay, still we pray. Okay, then any situation goes on. Okay, as for me, I thank God once again for giving me this golden opportunity to deliver this useful session, to share my knowledge among my fellow national, international participants, students, and young researchers. Okay, so this is what we are going to discuss in today's class. We will discuss about the biological data in digital symbol sequences. Okay, it's not about pure biological sequences. We are going to discuss in digital symbol sequences, and then whenever you have to process with digital symbol sequences, you have to collect the information. This information you are storing it in the database. Okay, so we discuss about the database annotation quality, and then we discuss about the sequence profile. Okay, the digital sequence profile, and of course I told you we discuss about the genomes, the diversity, size and structure, and then about the gene content in mammals. Okay, what you call it to be the human beings, and then we discuss about the genome as well as the other genomes. Yeah, please come. Yeah, and then we have the independent work also. Okay, of course we have some short videos to discuss the knowledge in our topics. So this is what we are going to discuss with you. This is the image of what you studied in your biology. You remember DNA? Okay, so this is the biological data in the digital symbol sequences. What we are going to do, we will have some chain molecules here. So definitely with the living organisms, definitely for the living organisms to evolve, definitely we need some reproduction process. Okay, right? So that will be in the form of the digital symbol sequences. Okay, digital symbol sequences. So here, if you have already studied about DNA or maybe RNA or maybe proteins, we have like amino acids, and then we have some nucleotides even. So definitely in the physiological environments, you can see the structure being growing. Okay, so whenever you have these digital symbol sequences, definitely like maybe you will have like a spiral like this one. Okay, so these spiral like sequences, definitely it will contribute to the the evolving nature of the living organisms. Okay, whenever they test for a human body, what they will test? They will check for the DNA. Okay, so sometimes our DNA might match with our ancestors. Okay, maybe if you take in the case of the the Chinese culture. Okay, Chinese culture, maybe some of the human beings in the 21st century, if they check the DNA, it might match the DNA of the ancestors. Some they might get some powerful qualities. Okay, maybe an art of fight, or maybe an art of protection against harmful diseases, the way they take the food. Okay, it all depends on the DNA. Okay, so DNA is very very important for the evolving of the living organisms. So I'm talking about the digital symbol sequences. So it is almost like digital in nature, but it's quite different from the scientific data that we are collecting. Okay, maybe like we have physics or maybe any experiment that we are conducting, but this digital nature of this genetic data is quite different. And as I told you, we had like amino acids, and then we had nucleotides which are in DNA. So definitely they are going to carry out large scale DNA sequencing. And with regards to the experimental process that we carry over with regards to relation in characteristics, or maybe location, or maybe ethics, several genome projects have been carried over. Okay, I hope you understand the word genome is nothing but gene related, genetical. Okay, genetical characteristics of a living organism. So several projects have been carried out with this small scale DNA as well as large scale DNA sequencing. Okay, in order to analyze the structure and in order to find the ancestral properties, parent properties. So several projects have been carried over. Not only like it's all about like direct protein sequencing with regards to location, with regards to relation in characteristics also, they are doing several more projects. Okay, and even for this one they have come up with several algorithms have done in the computational point of view. Okay, so our goal is to identify its molecular structure and function, and definitely we will have like different variations in the polymorphism. Polymorphism. Polymorphism means similarity in the characteristics. Okay, for example, if that person and this person, okay, maybe
their
characteristics, the way they think, the way they perform the actions. The twins or maybe triplets have some similarity in the characteristics. Okay, yeah, polymorphism. Okay, so this sequence patterns that we are taking into, maybe it will be potentially it will sound to be little bit noisy means the characteristics are the effect of the characteristics are too much. Okay, and then the variability resulting, we will have like random events even. Okay, so this is the database annotation quality. So the biological sequences, as I told you, they are digital sequences. So which means it has to be stored in the electronic version. Okay, the digital version. Okay, whenever you tend to program any languages, then immediately you use the computer, immediately use the software for programming. Okay, similarly for processing these biological sequences, we have to go with the electronic storage of information. And this is what the specialists from bioinformatics as well as biologists with the computer knowledge are preferring to. Okay, they want everything to be stored as a digital way. Remember in the earlier version, whenever you have to do something, immediately they carry a big some equipment and do some stuff. But now everything is being done electronically. Okay, right, you are you're getting, for example, if a woman gets pregnant and you can observe the baby moving, everything is being done electronically. Even during the COVID times, you need not have to go and visit the doctor. Okay, so these results are available with the surgeon or the nurse, so they tend to show to you electronically. Okay, maybe via the video call they will try to show you. Okay, even the doctor need not have to come directly to the hospital. Okay, he or she can directly see, can monitor the status of the patient just via the video call or by accessing a public data. Okay, so this is handling of information. Definitely we can do it in the electronic means, but still you cannot assure that you get 100% success with the instruments that we are using. That sometimes like we have the instrumental error, but the instrumental error is not very much. Okay, there is some tolerable limit, right? So the human brain, which generally we are gifted to have like a human brain, because human brain, it will be performing like millions billions of instructions even, and also with the the lifetime of the individual, like we live average maybe like 70 years, maybe 60, 70, okay, maybe 80 even. Okay, so these information is saved as content addressable schemes. Okay, so which means we have some past experience as well, we have some present experience as well. Also our human brain is able to perform better in comparison with a computer or maybe a electronic version of any other device. Okay, so here in contrast, okay, when you take in the application of the computing or maybe information technology, generally the computers, we can handle large number of numbers, which maybe the human brain might get tired. Okay, after 8 hours of continuous work, human brain gets tired naturally. But the computers, with regards to different networks or maybe across like different countries, it is still, it is performing better. Okay, also the content addressable procedures normally that is stored in the human brain, that the computer might not understand. For example, okay, today I have come up in this particular class. Okay, now I say to you, okay, maybe I find maybe two students regularly talking among themselves. Let us take in the case. Okay, now I say to those two students, don't talk. Okay, maybe next time I should see you not talking. Okay, next class I am coming once again. I see those students. Okay, then I see, okay, they are not talking. Okay, then maybe after some time if they talk, I warn them. I warn that particular student. Okay, so this is all about the previous experience that is actually stored in my memory in the human brain. But the computer will not be able to understand this one. Only if you try to feed this one that these two students are not are always talking means, so it has to be fed every time. So our computers, it's not like content addressable procedures for annotating as well as retrieving information. Okay, so that emotional characteristics, emotional attributes is not associated with this computer. Okay, right? Maybe the examples of maybe the names of the student or maybe what profession they are doing. Okay, it has to be everything fed into the computer. Okay, maybe you would be you might be a student alone, maybe you might be a student as well as a part-timer. Okay, also maybe your eye color is different, your eye color is maybe brown. Okay, maybe his eye color is blue, let us take in the case. So that differs. Computer will not be able to intelligently classify, okay, this particular student, this one, you have to feed it every time. Okay, right? So the present day computers definitely will have all these features. Also the biological sequence retrieval algorithms that we have over here, we will have like different sets of algorithms that we are feeding with. Maybe it will have like fuzzy based approaches, fuzzy k-means approach, fuzzy c-means approach. We will have different probabilistic functions in these retrieval algorithm. I hope you understood probability. As simple as that. If you take a coin, if you take a simple coin, you toss it, you toss it, you get head as well as tail. You toss a coin.
Which?
Head or tail, 1 or 0. So this is what you call it to be the probabilistic methods that you tend to use in the fuzzy representation schemes in the retrieval algorithm. So whenever you are performing these types of procedures in the laboratory, definitely it is stored as separate functions, maybe like maybe the marks or maybe the coloring or maybe like scribbles that you do it or maybe save it in the sequence. Okay, right. So if you take in the case of human visual systems, so definitely even in the brain, even in the sequencing of the organisms that we have in the human body, still it is subjected to error. Okay, so this errors are actually meaningless when you take in the case of the coding as well as non-coding regions in the genomic DNA. Okay, so what I can generally do that with the for the bio informatics specialist point of view, that we have to taken forward the errors, the amount of error, what is the error percentage, what is the tolerance level that we have to calculate from this ML approach. ML is actually machine learning. Okay, machine learning based approach with regards to prediction as well as classification. Okay, so still in the sequence driven mechanisms, as I told you earlier, in the DNA sequences we tend to use nucleotides as well as amino acids. From this it has been made compulsory, but still it is every time it is subjected to error. Okay, so the role of the bioinformatics specialist should be that it should be removing the error out of it. Okay, so that's the major decision or maybe the responsibility of the informatics specialist. Also, whenever you try to find out the error, okay, then you have to properly tell your other specialists also, okay, that there is an error in this particular part. Okay, sometimes what happens is we find something, we understand something, and then we say something. Okay, and then the others will not understand it. They will think, oh, okay, this is the error. Okay, this what you call it to be the annotation error. Okay, there might be error in here, there might be error here. Now I understand this is actually this one. Okay, okay, now I say this is the error. Okay, now my colleague, okay, when I say this to my colleague, he understands the error is here. Okay, so this is what you call it to be annotation. Everything is black. Everything is black, but he or she tries to understand that this is the error. Okay, but actually this is the error. Okay, so this is very important with the case of the machine learning algorithms. So whenever you talk about DNA sequences, it should have like the glucose as well as the phosphate levels to be moving in the right concentrations. So for correcting all these sequences, maybe we should perform some security checks. We have to perform some regular checks in order to understand these mechanisms. Okay, so that's the reason we go for fuzzy statements, where we generally go with evidence based approach. We generally calculate a experimental approach where we tend to involve some probabilistic functions. Okay, so where we tend to make the prediction analysis even more easier as well. Okay, so without proper treatment, definitely this problem is going to increase. So generally we are using these machine learning based approaches in this DNA sequencing. Like in the diagram, if you see over here, we had like different DNA sequences. Each of them we can differentiate with the case of the like normal adults or maybe a small kid or maybe a middle level human being that we can do so. Okay, so maybe it can detect this the language capabilities, like the infants, they speak ta 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 ta. Okay, now maybe a student of class 1 or maybe class 2, he or she will be able to speak a little bit better. Okay, so this recognition of word, this continuous speeches, it will try to recognize this. Okay, so since this language of the infant, okay, is unknown and it is complex as that of the DNA sequences, we need some learning techniques in order to better get this information in the case of genetics. And then there is database redundancy. So there is another problem, which is nothing but the redundancy of data. Redundancy of data is repeating of data is being repeated. Okay, so many entries in the protein or maybe genomic databases, we will experience this with the genes of similar characteristics. So what you call it to be the homologous genes, which are found in different organisms. So sometimes what happens is the characteristics of the homologous genes are repetitive. Okay, so it's this repetition causes a error. Okay, and also like we have like several groups, we will have like more or less closely related information that may not be similar, but they are considered to be identical as well. Okay, so for these things which are close to identical, so this it would reflect the genuine organism or the tissue. Okay, so here with the case of the DNA sequences that we use, so maybe it can be in the form of this one, cDNA complementary, which is just the opposite of the RNA which is present in the cell. So in the eukaryotes that you try to evaluate, we can find that the cDNA sequences, maybe the complete DNA sequences, will be having like premature form of the RNA sequences, and this genes, maybe what you call it to be the genomic DNA, it will combine with this DNA sequences, which are not continuous in nature, with the chromosomes of maybe a dog or maybe a cat or maybe like any mammals. Okay, so it will be having like slice based approaches where these genes may not be same. It will be individual with respect to each of the living organisms. So this is the example of the alternating splicing in the eukaryotes. Okay, so here we will be having like large number of genes which are very similar. The characteristics will be very similar, but definitely we will have like better reliability in the data collection procedures. Okay, so since we are able to collect the information reliably, okay, so these can be converted into data sets and it can be stored in the databases. Okay, so in the case of the mammals, or what you call it to be the human genome, so it is at least 30 to 80% of the genes are alternatively sliced. So this is what you have over here. See in the second over here. Okay, I'm pointing to this one. So it's alternatively sliced with respect to each other. So which means that 30 to 80% of these genes are alternatively sliced. So these genes, maybe it can be spotted into the glass plates whenever you are doing any experiments. When you tend to evaluate with the glass plates, these can be easily synthesized. It can be easily synthesized with respect to the DNA that you are observing, and then it can be converted into sequences, maybe a set of digital sequences. And these digital sequences, you can better take this for analysis even, and you can compare whether this DNA has a similar characteristics of the parent or not. So for this thing, we are embedding this computer processing information technology here. So here we will be using like micro arrays or maybe chips. We will have like more sequences where maybe definitely the quantization levels will definitely match into when the quantization levels match, definitely we will see a similarity in the characteristics. Okay, so the protein databases that we have over here with the case of the amino acid sequences, definitely it does not correspond to the direct translation of the wild type sequence of nucleotides. So generally we will have the data set to be having a good future of the sequences. Whenever you go with any experiment, you should go with calibration, testing, and then validation of procedures. Okay, so with the case of the digital sequences, generally the performance, it should be tested, even it should be validated even, and if it is reflecting the input with the corresponding to the output with the economy in the par, definitely this method is found to be really good. Okay, and that is the reason you tend to use the machine learning approaches, okay, the artificial intelligence AI based techniques over here. So even with the processing of the digital sequences, the machine learning algorithms, we are having some errors, we are facing some problems with it. Okay, so we should maybe work on collection of the correct data set, so it's not about every time the processing we have the error. Okay, so it is all about the data sets, how accurately you get the data sets and you can process it efficiently. So this is where there is problem in the machine learning based approaches. With the case of the protein structures that you are trying to evaluate, the data set that you are trying to get, it should be reliable, even it should not be a repetitive one. Okay, there should be a good redundancy in the data sets that you are trying to collect. Okay, so also several cases the problem lies with the redundancy, like whenever you are getting some output, that output we will have maybe like 1% or maybe 2% error, so that thing you should reduce. If the error rate, if the percentage of error rate is reduced, then we can improve the analysis procedures. So that's what we have discussing this many times with the prediction as well as classification based approaches. Even this data which will be full of error, it is very difficult to process. Okay, and this is where the machine learning engineers are facing problems with the analysis. Whenever you have large amount of data, okay, maybe this data might be prone to error. Okay, so this is the major problem with the digital sequences. So we have to focus on reducing this error. Okay, and also that, just as I told you with an example, the prediction of the wrongly assigned features, we will have wronger evaluation, and definitely the annotation features, what I am thinking, I am understanding in another way, and I am telling this in a wrong way. Okay, so these all contribute to the errors. So next we will go with the sequence profile. Okay, so it's all about relation with the sequence retrieval procedures with regards to alignment. So how you try to give the input and how it should be aligned, and then how it should coordinate to produce the output really matters with the sequence profile. So while this profile is no longer continue getting the information about the sequences with regards to individual means, so it's if it is very unique, if it is very powerful enough, so definitely these profile, it can be very much updated. Okay, for example, if in this class, okay, if in this class one particular student is very smart, okay, very smart, okay, and other students are very much dull, means that student can be updated with the newest technology. Okay, so that's what is similar to the sequence profile. So one particular profile is very powerful, means you can update it and you can make this profile to be the best one. Okay, the best one. So for this base the sequence profile, we have like different approaches, maybe like a Gaussian based mixture model, or maybe hidden Markov model that we can evaluate to identify or segment the region. For example, let us take in the case in the brain, okay, or maybe if you take in the case of a dreadful disease called cancer, okay, right? Maybe if there is cancer in the liver, let us take in the case, we first of all identify the liver, and then within the liver, after segmenting that liver, then we identify the tumor. Okay, this tumor might be, maybe let us take in the case if it is in gray color, let us take in the case, this gray color might be similar to the background, might be similar to this background. Okay, so you have to carefully analyze that this is the background and this is the tumor. You are getting? So this is where sequence profiling really acts, and you should be better able to analyze with the help of the different models, maybe Gaussian mixture model or maybe hidden Markov model as well. So that's the reason you use neural network. Okay, now coming back to our topic, most of the topics in this subject we will have genomes and proteomes. So genomes, I told you, it is the similarity in the characteristics of the genes. Okay, so the genomes of the living organisms will be having like diversity. As I told you, it can be single stranded DNA, single stranded RNA, or maybe double stranded DNA, double stranded RNA with respect to mammals, with respect to cat or maybe dog or maybe any living organism. Okay, so some genomes can be linear, maybe in the case of mammals it is linear, maybe with the others maybe it might be, it is not linear. In the case of the mammals, our genomes are linear, the characteristics are similar. Okay, characteristics are similar, it is not exactly the same. Okay, because the genomes vary from one human being to another human being. Okay, and with the others, even with the case of maybe virus or maybe any bacteria that is associated, it is circular or maybe closed. Okay, so here we, in the case of the cellular genomes, generally we will have like a DNA, but with the case of viral genomes, that we will have DNA and RNA. So the image on the right hand side, it is actually viral genome. Okay, so here we would have the information maybe in the positive sense, even maybe even the negative sense also, or maybe both positive sense as well as negative senses. Okay, so they have given some specifications. Okay, so that you can note it out. Maybe the positive direction, maybe it will be from 5 to the 3 end of the molecule, maybe it will be in inches, 5 inches to 3 inches end of the molecules. Okay, the smallest genomes maybe we can find it in like bacteria or maybe like viruses, and they have evaluated this criteria maybe during like 1970s. Okay, so that was what has been evaluated, and we find out that the small genomes will be having like continuous sense of characteristics. Okay, so the much larger 3310 megabyte per human genome, generally we will have like 22 chromosomes, and then we will have the another one like we have like X and then Y. Okay, the combination of, you remember this one with the case of biology, XX, XY, that combination only decides the type of the living organism and what type they will give birth. You can understand that pretty much. Okay, so even the monkey, what you call it to be the chimpanzee, have 23 chromosomes in addition to the 2 reproduction chromosomes they have. Okay, 2 reproduction chromosomes. Okay, I don't mention that 3-letter word, I talk it as reproduction organism. So in addition to the 2 reproduction chromosomes, we have 23 chromosomes. Even we can take in the case of this chimpanzee somatic cell nucleus, it will be having like 48 chromosomes, in contrast to maybe in the man we will have 46 chromosomes. Okay, maybe if you take in the case of the dog or maybe cat, dog, it has 78 chromosomes, whereas the cat has 38 chromosomes. So it is all about the based on the identical copies of the DNA that you can analyze the characteristics. Okay, so this is the a statistical way of analyzing with the case of mammals, birds, amphibians, fish, insects, plants, fungi, or what you call it to be maybe like bacteria and viruses even. Okay, so here we observe that it goes on, it increases. Okay, from like a viruses it might be 10,000, 100,000, 1 into 10 power 6, 1 into 10 power 7, it goes on. Okay, even in the case of mammals we have 1 into 10 power 10. Okay, so this is the intervals of genome sizes. So for mammals it is the highest. So the chromosome level itself is not stable. That's what I told you. Like, well, with the case of bacteria and the virus, generally we will have like stable component like 2.4 mbp, and maybe less stable component also 1.2 mbp, which can be observed in the characteristics. So these genome sizes we will have like maybe non-overlapping intervals, which means that they do not fit into certain characteristics. Maybe one type of living organism will be exhibiting one type of characteristics which is different from the genome size of a different level organism. So the genome sizes also has an interval change from 3.5 to 280 kbp, and the bacteria will be having like 0.5 to 10 mbp. So they are giving some examples like for fungi, what about the size, and then for plants, what about the size, for mammal, what about the size they are giving. Okay, so for each of the living organism, what is the genome size they are giving. Maybe for the bacteria it is from 0.5 to 10 mbp. For fungi, greater than 10 to 50 mbp. Plants means it is greater, again 50 greater than 50 mbp. Mammals 1 gbp. Okay, right. So one example, we would go with the enzyme luciferase that I would be showing you in the next slide with regards to different class of organisms. But what you can do is whenever you observe the characteristics maybe of movable organisms like maybe human beings or maybe birds that fly, like crocodile, maybe reptiles that you identify, they have some genome sizes which are to be more or less similar. Okay, maybe if you compare that with the maybe the virus or maybe the bacteria, it might be falling into one small kingdom. We all fall into one small kingdom. Okay, so as it is possible to estimate the size of the unsequenced gaps, definitely the size of the human genome is known with a high precision. So organisms definitely will need the same molecular apparatus even for calculating this genome sizes, and we will have different genome sizes that I can show you with one type of fish. Okay, this you call it to be the puffer fish. Okay, so as you can see, it will be like big bulge in shape. Okay, so this is actually belonging to Fugu rupreps genome. Okay, so that you can observe this with the puffer fish. Okay, it is 4 times larger than the normal genome size. Okay, like in the in the type of birds or maybe reptiles, but this puffer fish genome size, it's quite larger. Okay, okay, so that is a interesting category that you can observe, and for this the genomes range would be very much high, like 700 mbp to 80,000 mbp it might range to. Okay. These genes definitely it should align, it should work in coordination with the protein products. So the DNA as well as the RNA sequences, the data sets should be properly analyzed even. Only then the genome sizes will remain constant. So this is the independent work. I have already given them in the Google Classroom. Please complete them. Okay.